Hi, my name is Maria Marco. I'm a professor in the Department of Food Science and Technology. It's my pleasure to welcome you to this Wellness Academy, and I'm really excited to share this time with you to talk about our microbiomes. And what I mean by this and how I think about it would be our old friends and our old friends that provide new opportunities for health and wellness. But let's really frame this so that we have a perspective of where we stand with microorganisms. We as humans and our ancestors have really only been on Earth for less than 1 million years. And while that may sound like a long time, it's really not. Microorganisms have been inhabiting our planet for approximately 3.7 billion years, which is about 4,000 times more time than us. So perhaps it's not really a huge surprise when I tell you that microorganisms are essential to the health of our soils, our plants, oceans, lakes and rivers, the atmosphere, and of course, animals and us. We've evolved to be with microorganisms on our body. And not just a few, but many. At this time, each one of us is really less than 1% human. That always blows my mind. What I mean by this is that if we add up all the genetic contents of microorganisms on our body, they are 99% of us. That number goes down a bit when we talk about the number of bacteria on us. We're at about 1-to-1 or 10-to-1 bacteria to human cells, but that's also not trivial. And if my point still isn't clear, right now, each one of us has more microbes on our bodies than there ever have been people on the planet. Yes, not us collectively, but individually. So I'm going to be pretty biased here in my conversation with you and talk about bacteria. But before we go further, I think it's important for all of us to understand that our microbiota, or in other words, microorganisms that are defined as a collection of all the microbes in our defined environment, the microbiota, is comprised of bacterial cells and other microbes like fungi in the form of yeast, protists, including humus, and other eukaryotes that are not harmful, and viruses, a very large group of, microorganisms that aren't alive, but we categorize together with our microbiota because of their small size. So we put them all together, we're talking hundreds of trillions of microorganisms. But as I said, I'm going to be focusing on bacteria because they are the most abundant group that can be easily monitored. We simply don't have the right tools available yet to fully comprehend our fungal, eukaryotic, and viral microbiota. We're getting there, but I'm going to focus on bacteria for today. Also important to understand in this conversation is that much of what I'm going to tell you is a result of significant advancements in our capacity to understand microbial life that have happened over the past 20 or 30 years or so, including genetic analysis and computational power to investigate microbes wherever they may be. So with that said, we can now have a picture of the different kinds of microorganisms, and again, bacteria, in different environments, and including our bodies. So fortunately, what we've learned is that the bacterial composition on our bodies is, in some level, not very complex. At a very high taxonomic level, called the phylum, there's only a few, 6 or 7 different phyla we think that are really important for our human microbiota. And that is in reference, if you look at the big picture, there's estimated to be over 1,300 different bacterial phyla on the planet. So 6 we can work with, not terribly complex. And by looking at the different phyla, we can see in different sites of our body that they come in different proportions and different abundances depending where we look. So we'll have microbes on our skin, in our nostrils, in our genital tracts, in our intestines, every exposed site on our body. And yes, even in our digestive tract, which seems like it's inside, but still technically the outside of our body. But the picture gets more complicated. As we go deeper into taxonomy, beyond the phyla to class, order, genus, species, we can look at the different species of microbes on our bodies. And there, as I mentioned, it gets more complicated. There's thousands of different bacteria species in our microbiomes. And we could look at it very simply if we take this picture of this football stadium and all of the fans that have organized themselves based on their team and different colors. And I would suggest we look at them as different species. Depending on the color would be a different species. Why do I ask you to do that and not phylum? Because there's actually more complexity than species. And so what I want to make sure you understand as we move ahead today is each of us not only has different species, but different strains. Strains would be sort of the deepest taxonomic level we could go in looking at microorganisms. And really, there's just an infinite number of strains in the microbial world. So going back to this image of this football stadium and all the fans, we would say that each person there would be a different strain within our human species or in the bacterial species in the different colors shown. So because of this diversity and taxonomic diversity and in fact genetic diversity, not too long ago it was proposed in a very elegant paper that our microbes or our microbiota are so stable and personalized between us that they can be used as fingerprints. By this I mean, just as we have different fingerprints between us, we have different microbiota. And they're so consistent throughout our lifetime that they could tell me apart from you. Okay, so we have this really diverse collection of microbes on our bodies, and told you a little bit about that, and then we can kind of wonder, well, if they're so different, if we're so different from one another, how at all could they be doing something similar between us? And in fact, they do. So on the left here, you can see this kind of species, or we could go to strain, but really species overlap between different people at the top. But when we start to look at what enzymes or metabolisms or qualities they encode and impart on us, there's a lot of similarity. So that would be the functional overlap. And this is shown in a different way in the figure on your right, in that we could look at the different phyla again on the left, and there's a lot of there's several different colors, but different proportions of different phyla, we would add them up. And of course, that would be extremely complicated if we looked across different people. And again, just to reference you, each of those little columns would be encompassing hundreds of different people. And on the right, we take the genetic contents of those different microorganisms, and we look to see what genes they encode and different enzymatic capacities, and a very different picture starts to come out. And that we see these very consistent colors across different individuals, and each color represents a different enzymatic function, and showing that even though you and I have different strains or even different species of microorganisms inhabiting our body, those different microbes have we've adapted, or they provide a specific set of capacities, enzymatic, metabolic, and so on, that are going to be really impacting us. Okay. So this lays the groundwork for where we're going, and that is to our digestive tracts. Why the digestive tract? Well, it's because it's the site in our body that has the most microorganisms. I probably don't need to go further than to say we excrete our own body weight in fecal bacteria annually. That's sort of a mic drop moment for me, just to think about the number of microorganisms inhabiting our digestive tract, in particular our large intestine or colon, right now. So let's take a quick tour through the digestive tract. Of course, we start with the mouth, which just has its own set of microorganisms and is very important, and I'm not going to talk about it further. We go down to the stomach, the small intestine, and the large intestine, and it's a whole world for microorganisms in there. Of course, there's the food or beverages that we consume that are going to affect our microorganisms, and I'll come to that in a moment. But our own physiology, our own adaptations, our evolution have created a system that creates different microenvironments for microorganisms. So we can look at how the oxygen changes, more oxygen available for microbes to breathe at the start of the digestive tract, and that goes away towards the end. And we have the kind of microorganisms that thrive without oxygen present, or anaerobes. We see a change in pH that's needed to digest our food, but the microbes have adapted to these different pH conditions, from very acid in our stomach to more neutral pH conditions as we move towards the colon. There's antimicrobial peptides, there's bile acids, there's short-chain fatty acids. All of these different compounds affect not only our own bodies, but also the microorganisms. And then I'm going to take you to the numbers of microorganisms and the different families that we would find there in these different sites. So as we go down our digestive tract from our stomach, which has microorganisms, it's not devoid of life, microbial life, even though it is very acidic. And we go to our small intestine, and then to our large intestine and colon. And so what you should see is those numbers of bacteria, or expressed in log units, would be CFU is number of cells approximately per gram. We get up to 1 trillion bacteria per gram. And then we'll see the same kinds of phyla and species and families that we saw earlier, with Bacteroides, Firmicutes, and Proteobacteria being the most dominant. Okay, so this is the tour of our microbial world, or on our bodies and in our digestive tract, but we're here to ask, how do these microbiomes affect our health? And some of this we've known actually for quite a long time, but we're still learning. So let's think about how these microbes are good for us, and please keep that in mind, since we have evolved as Homo sapiens, as humans, to be together with our microbiomes. So, our microbiomes are really important for digesting our food. Our own bodies can only make a certain number of enzymes and only take a digestion of our food so far. But the microorganisms inhabiting our digestive tract, in particular our colons, are well adapted to continue to break down our foods and fiber, for example, releasing important nutrients that can improve our health. Vitamin synthesis, amino acid synthesis. So microbes in our digestive tract make vitamins we cannot. They also provide amino acids that our bodies do not make. Microorganisms in our digestive tract are important for food and drug detoxification. A lot of this we're still learning, but there is connections between the types of drugs we're taking and the microbial metabolism and the efficacy of those drugs on our health. Pathogen exclusion. If you ever taken antibiotics, you may have had some antibiotic-associated diarrhea. And that is in part the result of the antibiotics not only killing the pathogens, which for the disease we need to treat, but also diminishing the vitality and the numbers of the good guys in our digestive tract. So we need to have a fully colonized, healthy microbiome to exclude external pathogens. And then we're getting into other ways microbes are good for us, in ways that our imaginations are really just starting to take off with, in not just our digestive tract, but our overall bodily metabolism, the function of our immune system, and importantly, how our gut and our brain are connected through the gut-brain axis in altering our moods and our behaviors. So hand in hand and understanding how these microbes are good for us, we're starting to think about exactly how they do this. So what are the specific metabolic or enzymatic potentials that these microbes carry that are really important for what we get out of them? So it can be through any number of protein, metabolite, lipid, carbohydrate types of metabolisms that these microbes are resulting in the benefits that we see. Okay, but it's not all happy news. The more we learn about the associations of our microbiomes and our health, the more we're asking, well, could microbes inhabiting our body also harm our health? We clearly see that with pathogens, of course. Viral pathogens to bacterial to you name it, right? External pathogens that we come into contact with. But what about our own microbes that are on us right now, which we don't really see or maybe feel healthy? We don't have a problem with. They seem to be functioning well. But there could be some issues lurking beneath that so-called healthy microbiota that we need to explore. So for this, we sometimes say gut dysbiosis. Okay. It is a term to imply that we're not always colonized with the good microbes or beneficial symbionts, but microbes that would be pathobionts or harmful in certain situations. So it can be the presence of those pathobionts or the absence of the good guys that can cause disease or make conditions worse. And frankly, I could spend the next 20, 30 minutes describing to you all of the different studies that are out there associating our microbiota composition, in particular gut microbiota composition, and disease. There's many, and it's not just limited to effects on our gut health, like inflammatory bowel disease, or gluten sensitivity, or GI transit times. The connections between our gut microbiomes and our health go well beyond that to associations with development of allergy, type 2 diabetes, type 1 diabetes, depression, and the many other conditions you see here on this slide. So while many of these are associative, and there's many questions as to which microbes or why and how, we are rapidly learning which one of these conditions and which microbes are important for causing these outcomes rather than merely associated. And through the exploration of our gut microbiomes and this connection with health and disease, there have been numerous studies that have tried to make these connections of what factors in our lives determine our microbiome composition. And in this very colorful slide, you can see many different measurements individually and collectively being shown. And one take-home message of this is that a lot of it still cannot be explained, about 85%. But of what we can explain, what determines our gut microbiome composition, we find that medication, so drugs, determines our microbiome composition, our body weight, our existing conditions are associated with our microbiome composition, and importantly, and if you look carefully at the very colorful chart on the left, diet. Diet determines our gut microbiome composition. And it's this last one that gets me most excited because it is actually something we can do something about. Okay, so there are many different factors we are seeing that are associated with the composition of our body's microbiomes and in particular our gut microbiome. Again, kind of represented graphically here. So with that, it's really inspired many different groups of people to look to the microbiome as a prophylactic and therapeutic target, meaning we can use our microbiome to prevent disease and we can use it to treat disease. And as we think about that, we're trying to tip the balance away from any harmful microbes to those that would be beneficial or non-harmful, called commensal. How can we do this? Yes, we can invest in more drugs. We can also use tools like fecal transplants, which is a really important therapeutic for individuals, for example, who have Clostridium difficile infections of the GI tract. So really important tools, but what we expect you'll need a prescription for. But there's still diet. We need to eat and drink every day, and so this is something we can, in part, have some control over. The importance of diet on our gut microbiome has actually, it's kind of been known for a while. And all of these sort of epidemiological studies connecting microbiome composition with different anthropomorphic and dietary factors, there are these connections. But some of the early studies on the gut microbiota showed how different the colonic microbes are between individuals, so-called a plant-based diet, and those with an animal-based diet, shown by the different colors in the pie charts you see here, and at a kind of a higher level, kind of different types of microorganisms that really stand out depending on what diet is being eaten. Again, plant-based carbohydrate or animal-based high protein and fat. And going to be the one to deliver the news that really just having a healthy diet is going to not only help you, but also your microbes, and cultivate the kinds of microbes you want to have on your body and in your digestive tract. So it's not really a big surprise, I think, for us to hear that staying away from sugary foods, snack foods, fried foods not only helps our health, but also helps the health of the microbes, which in turn help our health. Probably the best example of this benefit is through fiber, dietary fiber, which if you ask a nutritionist, it's still really hard to define what dietary fiber is. But I think we get a general idea of having, whole wheat bread, oatmeal, kind of different kinds of fibers in our diet. And just as this headline here shows, high fiber diet helps treat diabetes by changing gut bacteria. Really nice paper published in Nature that year. And they aren't the only ones. Many others have found that fiber intake is overall associated not with good health, but directing the microorganisms in our intestine towards that direction. One example from my own research is looking at a type of wheat that's enriched in a dietary fiber called resistant starch. And in the types of nutrition studies that look at diet, we investigated people that had a diet enriched in this resistant starch wheat or their regular diet, and it was crossover, so they didn't consume these diets for very long, or the addition of the resistant starch wheat for very long, for it was only about a week rather, and what you see is a very rapid change in that the resistant starch enriched wheat reduced glycemic index and improved insulin responses in this small study. As I said, this is just one example of how diet, in particular fiber, is able to fine-tune our microbiota in the direction we want to improve our health. With that said, other studies that we've also published have shown that it is still a bit of a question on exactly how and which microbes are being enriched and what the benefits they are that they provide. So even with fiber, I feel like we're still scratching the surface of where we're going to understand the connection between our microbiota and health. And this is illustrated, kind of the issue is illustrated here in another study that we performed, and this was of the high fermentable carbohydrate diet, so a high fiber diet, where each one of these lines would be a different person consuming either this healthy high carbohydrate diet or the green, or a Western style diet of hamburger and french fry diet, normal diet, in purple. And what you see is that some people had a very long arrow in the green, and that means their microbiota changed a lot when they were consuming this high carbohydrate diet, where others, you see the arrow was quite small, and the purple you see very little change, which is what we expected because they were basically consuming their normal diet. So while this change in some people was associated with enrichment of a good microbe called Bifidobacterium, not everybody responded the same way. So what this means is that, going back to our individuality, each of us have a different collection of microbes with different enzymatic potentials, and so some of us are going to respond better or enriching the right kinds of microbes in high numbers when we have a high carbohydrate diet or high fiber diet, where others, we may see less change. And we know that from looking at this kind of very complicated pie charts here on the right, the different enzymatic capacities differ between different people and their collection of microbes that they have. So there's still a lot of questions, yes, even with fiber, but the bottom line is it's not hard, it's not a bad idea to include more fiber in your diet. Okay, so besides fiber, what else should we be eating? And now I'm going to get a little bit provocative. So for about 25 years, there's been a theory out there that maybe we should be exposed to more microbes, not less. And New York Times featured this back in 2015. If you look at the New England Journal of Medicine from 2002, supplementation with lactobacillus, so one of those good guys. The dog at home, attendants at daycare are protect against the development of allergies. So this idea is encapsulated in a hypothesis called the old friends hypothesis, or the biodiversity hypothesis. In that, just as our lives have been benefited through industrialization and the many attributes we have for a good quality of life that didn't exist 100 years ago, there might be some cost to that as well. So just as we see a decline in biodiversity on the planet, we see a decline of biodiversity on us in our microbiomes. And so this idea of microbial deprivation or loss is related to this dysbiosis concept that I introduced earlier, and related to that, a disturbed immune response. And then we can get into a vicious cycle of increasing our risk for inflammatory diseases. And this idea has been tested in a number of studies, but the idea here signals that what we see from epidemiological studies kind of relates to this biodiversity hypothesis. For example, examination of the development of allergic diseases like atopic eczema, allergic rhinitis, it's really increased, or say exploded, over the past 30, 40, even 50 years. So that's allergy, and also another disease which is increasingly prevalent would be digestive tract related or inflammatory bowel disease, which also is just, in the number of cases, the number of individuals dealing with this issue has increased exponentially in recent times. So the past few years, myself and friends and colleagues have been kind of thinking about this idea of the hygiene hypothesis, or missing microbes, and looking at the foods that we're eating. So our earlier diets, and before industrialization, and certainly our distant ancestors, didn't have access to refrigeration or ways to preserve foods like we have today. And we're probably eating more rotten food than we would care to eat now. And with that comes a high diversity of microorganisms into our digestive tract. Another thing to point out at this point is our digestive tract is also home to the majority of our mucosal immune system. So any microbes that are in our digestive tract or coming in on foods are going to be sensed and recognized by our immune system. So then as our foods became arguably safer through food safety measures, and absolutely important point to make is that our foods are safer and we want them to be free of pathogens. But while making them safer and last longer on the shelves in the grocery store at our home, we've also removed microbes. So we may have lower exposure to microorganisms through our diet. And so we've been exploring the idea of what happened if we increase the number of microbes back, maybe not to our early ancestors, but back to maybe our ancestors from a couple hundred years ago were exposed to. So what are the sources of safe live dietary microbes? These would be some fermented foods. This could be probiotics, or it could be even the microbes we have on an apple or in a salad. So by fermented foods, make sure we're all on the same page. Again, it's a very microbial-centric topic in that fermented foods are foods and beverages made through desired microbial growth and enzymatic conversions of food components. So that's what a fermented food is, ultimately also microbial. And some fermented foods, as I mentioned, will have live microbes. Not all. For example, when you bake bread, there will be no microbes alive any longer. Many of these foods that we cooked, you could read down the list here, they're filtered or roasted to remove any microbes. So when we eat them, there won't be microbes present. But all the foods on the left, like yogurt, unpasteurized kefir, kimchi, sauerkraut, will have live microbes in them. And just as it's very humbling to think about the number of microbes on our body, we only have to look for a cup of yogurt to be reminded that there should be, and typically are, as many live bacteria in a cup of yogurt as there are humans on the planet. The other term I mentioned would be probiotics. This is another source of safe live dietary microbes. Probiotics are very specific microbes, strains, that when administered in adequate amounts confer health benefit on the host, us or animals. For probiotics, there's over 1,000 or thousands of human studies, mainly focused on treatment or prevention of disease in the digestive tract, but there are also a lot of probiotic trials currently ongoing to assess their capacity to help us at other sites of the body, from allergy to cancer to type 2 diabetes risk. Right now, so we're on the same page, these probiotics are mainly strains of lactobacilli and bifidobacteria, two good guys. So our question in our group, and this is research I'm doing together with the International Scientific Association for Probiotics and Prebiotics, or ISAPP, we're asking, do live dietary microbes improve our health? We don't have an answer to this entirely. When we put them all together, all the microbes be exposed to, but we are testing the hypothesis. So we're asking the question, what foods contain the most microbes? Who consumes microbe-rich foods? Can we find a link between microbe intake and health? What are the confounding factors to be considered? And should we be recommending more microbes? And if so, which ones? So to start to dive into this hypothesis, we've been using NHANES. NHANES is a database managed by the CDC. It's called National Health and Nutrition Examination Survey, and it's a cross-section of adults and children across the U.S. examined or asked about their diet, exercise, and measured for their health status. And each cycle, about 5,000 people are included in the survey, and it's a really important survey, in fact, for dietary recommendations and overview of where we are in the US in terms of our health. So we took a dive into this NHANES database, and in there we can find all the different kinds of foods people were eating at the time this survey took place. And we loosely grouped the foods whether they had low numbers of microbes, medium, or high. And you can see the numbers here. Low we put down to be if you have a glass of pasteurized milk, not going to be a lot of microbes, or if you have a slice of pizza, not a lot of microbes on there. Medium number of microbes per food, per gram, would be like a fresh salad or fresh fruit, vegetables. High would be fermented foods or probiotics. And what I'm putting up here are the percentages of the different foods people ate that fall into these different categories. And sadly, but perhaps not too surprised to me or you, anyone, that most of the foods we eat are processed heated and have no or very low microbes present, whereas only 1% we would put into the fermented food category. So what was some key takeaways of our first dive into the NHANES database and looking at dietary intake? What we found is only about 2/3 of the US population eats any microbe-containing foods. So there's many of us who really don't eat fermented foods or foods that would have any fresh fruits and vegetables, for example. Another key point, those that do eat live microbe-containing food, we get about 100 grams per day. And if we add up the microbes we estimate to be in those foods, that's about 100 million to 1 billion microbes per day. When we put into the big picture of the microbes in our digestive tract, one might say, that's not a lot. That's possible, but it could be that these microbes are making more contact with our small intestine where there's fewer microbes of our own present that can make a difference. Most of these microbes we would be getting would come from fermented foods, just in the sheer numbers that they would contain. So this next step that we took with the NHANES survey was not just to add up the number of microbes eaten or who's eating live microbe foods, but to associate the health data from NHANES with these individuals. So NHANES contains a lot of different health indicators, and for our purposes, we really focused in on looking at cardiovascular health. And in fact, when we did that, we found significant associations between metrics like body mass, blood pressure, cholesterol, triglyceride levels, and intake of live microbe food. So in all of these cases, or all of these metrics, consuming live microbe food was associated with a good outcome, meaning lower blood pressure, lower body mass index, lower triglycerides. And this is just for fermented foods only. You don't see an association with weight, but with total microbial intake we do, not just fermented foods. We aren't the only ones, so we are starting to find other studies and others taking interest in this topic, and showing here just a recent publication basically saying the same thing with NHANES, and that a high dietary live microbe intake was associated with a lower prevalence for cardiovascular disease, and significant association was detected for a reduction in stroke and heart attack risk. Okay, so there are some caveats here. Obviously, these are not just foods that contain microbes, but as I mentioned, fruits and vegetables would count as containing live microbes. So yes, there are some confounders that we have to work through. We know that fruits and vegetables are healthy for us in more ways than one. They also affect our intestinal microbiota diversity, so we may not just be counting the microbes on those foods, but overall the overall impact on our health. We're aware of this, and more studies are needed. But I do want to leave you with one more thought on these fermented foods. I find it amazing as a microbiologist who studies these foods is that according to dietitians, fermented foods are superfoods, and they've been so for 6 years and running at the #1 spot in today's dietitian poll as being the best foods you could be eating. And they're understood to promote good health. We see an uptick, interest in fermented food making at home and commercially. And so I encourage us as consumers to be kind of aware of which of these foods will be fermented, looking at the refrigerated section of our supermarket aisles where those microbes would still be there. And as we eat a fermented food, as we would eat a fruit and vegetable, there's more to these foods than the microbes. What happens through that microbial growth and activity in making the foods is not just the presence of microbes, but even foods that are cooked or roasted after fermentation, there's still going to be the outcomes of those microbial activities, including transforming the food ingredients, improving digestibility, and removing toxic substances, and synthesizing new compounds like vitamins or antimicrobials called bacteriocins. So just one example of this from my own work, and a talented grad student in the lab, Glory Bowie, we've been looking at how fermented food, and minus the microbes, just what they've made, can improve our gut health through our intestinal epithelium. And to say that our intestinal barrier, just like our skin, should be intact, and not letting microbes or foods or harmful compounds through into our body. So the better the barrier, the better our health. And so we can study this in the laboratory using cell culture and probing with different kinds of perturbations like pro-inflammatory cytokines. And lo and behold, when we look at the effects of a fermented milk as opposed to a milk alone, we see an improvement. We see an improvement on barrier function that's disrupted by inflammatory cytokines. Just put it all up there. That when we have cytokines present, we see harmful substance called fexidextrin go through the barrier. When we add milk, we see some protection. When we add fermented milk, we see even more. So it's not just the microbes in a fermented food, it's what they make, it's how they change the food that ultimately is going to be important for improving our health. Okay, in summary, our well-being is dependent on our microbiomes. Don't let anybody tell you differently. We could go on for hours talking about how our microbiome is good for us or associated with conditions, disease, or feelings that are as unwanted. And also related is our diet and the microbes in our diet, or the microbial products, for example, like ethanol and alcohol, that are affecting our microbiomes or affecting our health. I'd also say that we're in the era of the microbiome, and that it's really captured our imagination for both treatment of disease and prevention of disease and overall maintenance of health and well-being. Diet is really the way that we have in front of us as we think about what we put into our bodies. It's not easy. It's not easy to have a healthy diet, but it is something we can be adding maybe a little bit or changing a little bit that not only helps us, but also our microbiomes and our friends, and getting rid of our foes. So with that, I want to acknowledge Glory Bowie, who's center in this picture here, the numerous funding sources in my lab. You're welcome to look me up. And I'll leave you, it's just a microbial world. There's nothing else to say. Greg Venter put it nicely, if you don't like bacteria, you're on the wrong planet. This is the planet of the bacteria.